In this module, we'll be talking about light microscopes, which represent the vast majority of microscopes you'll see in a laboratory setting. In their simplest form, a light microscope consists of 5 different parts. First of all, you have a light source that emits light in all different directions. This light is collected and focused on the specimen with a condenser lens. The light then travels through the specimen and is again collected and focused by another objective lens onto a focal point that you can view. That produces an image such as the one shown on the right here. While simple light microscopes are sufficient for viewing colored samples, most biological samples, including cells, are transparent and will almost appear invisible as shown in the image on the right. Therefore, most simple light microscopes are upgraded with 2 pieces, a phase annulus and a phase plate. These 2 pieces work together to cancel out any light that passes through biological samples. The end effect is that any light that passes through the cell membrane is actually canceled out and appears as a dark line. This provides contrast that allows you to differentiate the cell from the rest of the surrounding environment. For example, you can see here on the right, the image on the bottom is much clearer and easier to see than the one on the top. This is an example of an inverted light microscope. It's called an inverted light microscope because the light comes from the top and goes through the sample to the bottom, instead of the classical setup in which the light source is beneath the specimen. To use this microscope, start by turning on the light by rotating the light knob. Then push the phase annulus into place. Next, place the specimen on the platform. Take off your goggles and look through the eyepiece to the sample. You may need to adjust the focus. This microscope has 2 focus knobs, a major and a minor focus, as shown here. Once you've got the sample focused, move it around the platform to find what you're looking for. You may choose to start with a low power objective, for instance 4x, and move to a higher power objective, 10x. You may then need to readjust the focus to clearly see your sample. Once you are done, turn off the light and put the specimen back into the incubator.